Hello, everyone. Welcome to December's EMSL Learn webinar, where today we'll be discussing the power of top-down and native proteomics for your research. We're glad that you can join us today for the EMSL Learn webinar series presentation, Realizing the Power of Top-Down and Native Proteomics. We have two EMSL Learn webinars next month. The first will be held on January 12th and is focused on tips for submitting a successful proposal. EMSL has two call for proposals open right now. So this webinar is designed to help you submit the best possible and most competitive proposal. There will be a link in the chat window for you on to the website with more information on these open calls. The second EMSL Learn webinar next month will be held on January 26th and is focused on characterizing atmospheric aerosols. We hope to see you there too. Also, make sure to keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. Links are in the chat as well. And this is a great way to keep up on dates for open call for proposals and other opportunities. So today we're going to begin with EMSL lead scientist for visual proteomics, Paša-Tolić, who will give an overview of top-down and native proteomics and the capabilities EMSL offers in this area. After Lily's presentation, James Umen, principal investigator at the Donald Danforth Plant Sciences Center, will discuss how he uses EMSL capabilities to identify core histone proteins and their proteoforms in green microalgae. We will end with EMSL chemist Mowei Zhou, who will showcase how mass spectrometry can resolve heterogeneity and lignin metal ion binding of protein complexes. There will be time at the end of today's webinar for questions. So we encourage you to post your questions in the Zoom chat window, and we'll have our speakers answer them during the Q&A. And with that, we'll begin with Lily.
Hi, good afternoon, good evening, depends on where you are on the globe. We are happy to have you with us and hope you are going to find the information we will present useful for your research. I will give you a very brief overview of the two topics that we will discuss today, and then Jim and Mowei will provide much more technical detail and examples for you to try envisioning how you can implement these technologies for your own research. I'm not sure I can. Okay. So at EMSL, we are primarily focused on post-genomic measurements, so primarily proteomics and metabolomics, lipidomics. And today we will discuss the proteomics side of the equation and specifically look at the intact proteins and protein complexes. Oops, okay. It's a little bit difficult to find the arrow to move forward, so sorry for being slow. So as you all know, proteomics is typically done using what we call bottom-up approach. So by digesting proteins into peptides, typically using something like trypsin, and then analyzing these peptides using some mode of chromatography coupled to mass spectrometry and searches against known genome information. So in this approach, we use peptides as a proxy for a protein, and it is an extremely powerful approach where we can very quickly actually characterize a good portion of a proteome, say 10,000 proteins from a human or something at that level. However, there are challenges, and I tried to illustrate that by this sketch below here, where you can see three different forms of the protein with three different post-translational modifications. So once we chop this protein into pieces, we get this mix of different peptides. And even if we are able to detect them all, which we typically are not, we have no way of assembling them back in a specific form of a protein that was originally in our mix. So to capture that information, we have to bypass digestion and characterize proteins using what we call top-down proteomics. So applying chromatography coupled with mass spectrometry to directly analyze intact proteins. Why is this important? Is illustrated on this slide, where as we go from the gene to the protein, there are so many different processes that can happen. And due to this post-transcriptional and post-translational regulation, a single gene can produce, and typically does produce, a whole set of different protein forms, which we today call proteoforms. So this protein diversity and complexity is actually fundamental for biology because it enables formation of distinct protein complexes or molecular complexes, and also establishment of different networks that drive biology. So knowing proteoforms gets us much closer to the function and the phenotype. Just as an illustration, and this is an example that is very familiar to all of us today, spike protein from the SARS-CoV-2 virus is typically decorated by sugars, and these sugars essentially hide the virus from the host immune system. Not only that, but they also, as illustrated here on the right, serve to enable protein to assemble in such configuration that it becomes possible for the protein and the virus to interact with the host cell. So you can see here these specific glycans kind of prompt spike protein into position to start interaction with the cell. So it's moving on its own. Similarly, for instance, glycans are extremely important for, say, activity and stability of different enzymes that are of interest to many EMSL users. For instance, enzymes that are important and are enabling degradation of lignocellulose material. And another example is histone research and epigenetics, which Jim is going to describe in much more detail. So as I mentioned, these post-translational modification and specific proteoforms rarely work on their own. They typically assemble what we call molecular machines, and these molecular machines are essential for cell function and biology. So in order to characterize them, mass spectrometry community has developed this unique and very powerful tool, native mass spectrometry, where now we are not working with just an intact protein, but the entire intact complex. And we can define this higher order structure, conformation, stoichiometry, interactions with different smaller ligands in order to better understand metabolic pathways and cellular and other cellular processes. So the next frontier that we have available to us then is to place these proteins and protein complexes in the context of the cell, as illustrated here on the left. So to define spatial distribution and orientation of each of the protein or protein complex relative to all of the other cellular components, and figure out how that spatial and temporal organization enable biology to happen. So we are planning to do this by integrating proteomics, and specifically top-down and native mass spectrometry with electron microscopy, single particle analysis, as well as electron tomography, also NMR and advanced computing and modeling, with a goal to develop this interactive Google Cell Atlas that will enable us to better understand cellular states and connect them to a specific phenotypes. So that's the future. Today, though, we are going to hear much more about methodology that we have developed to characterize histone proteoforms from Jim, who will describe how he applied these technologies for characterization of Chlamydomonas histone proteoforms. And then Mowei will take us on a tour of native mass spectrometry, if it shows up. And give you a couple of different examples from some recent work with EMSL users to inspire your imagination and enable you to think about similar or even more exciting approaches that are applicable to your research. So with that, I'm gonna give my control to Jim.
Okay. I'm not sure if I have control yet. Let me see.
Try to request.
I'm gonna do it again. Okay. Sorry. Okay, I think I have control of the screen. Hi everyone, I'm Jim Umen, and I'm just gonna tell you briefly today about a collaboration we have. We're an outside, with EMSL, I represent an outside user that really benefited greatly from having the expertise and instrumentation available at EMSL. The project I'll tell you about is a mass spec project, and we're not mass spectrometrists in my lab, but we have questions that really can only be answered with very specialized types of mass spectrometry that are available at EMSL. And so our work is a collaboration. It involves my graduate student, Sarah Rommelfanger, our EMSL collaborators, Lily and Mowei, and Jim Pesavento at St. Mary's College, who also has special expertise in histone post-translational modifications and mass spectrometry. Okay, so our experimental organism is the unicellular alga Chlamydomonas. It's a JGI flagship model. It's got a genome sequence, and lots of research is done on Chlamydomonas. This is a cartoon of what one of these cells looks like. It's of interest experimentally, not just as a reference for green algae, but also because it's a microbial eukaryotic relative of land plants. And it's also a good experimental system. There's well-developed tools, genome sequence transformation, gene editing, forward and reverse genetics, and indexed insertional mutant libraries. So this gives us lots of experimental avenues to follow up on things that we find using proteomics, and also to drive the experimental questions we're trying to ask. And so the interest in my lab is in the cell cycle of Chlamydomonas. It's a unique biological system for investigating size control. So Chlamydomonas cells divide by a somewhat unusual mechanism called multiple fission. And this cell cycle, the cells stay in G1 phase for a very long time and can get very, very big. So they grow more than 2 times in size during their G1 phase. And at the end of their G1 phase, they undergo a series of rapid divisions to produce daughters. And the general rule is that the bigger they get, the more divisions they undergo. And I'm just waiting for my next little animation to show up here. There it is. So the general rule is the bigger the mother cell gets, the more times they divide. So there's not a prescribed number of divisions. The division number is adjusted to mother cell size, and so they have this kind of biological counting mechanism where they know how big they are and convert that into the correct number of divisions. And we're really interested in how this biological sizer works. So one of the approaches we've taken is using genetics. Again, sorry, there's a little bit of lagginess in this, so there we go. And so before I tell you about genetics, I also want to mention that this is a great model system because it's easy to synchronize. So in light-dark cycles, these are newborn daughter cells, and they'll grow during the light phase, and then over a very narrow window of time, they'll all undergo that multiple fission and produce new daughters. And when my next panel shows up, it'll show you one way we've leveraged this natural synchrony. We can do transcriptome analysis. This is something we published a while back. The entire transcriptome, almost the entire transcriptome is produced very synchronously, and almost all of the core cell cycle genes are transcribed in a narrow window right around the time that cells divide here. And again, I'm not sure. There we go. When my animations are going to show up. So these are the cell cycle genes, and I was going to show in the next slide. This is a zoom in, just looking at the transcription of core histone genes in Chlamydomonas. So they almost all of them are transcribed in a burst here during cell division when they're needed. There are multiple copies of each one. That's why there's more than one line here. There's a few that aren't cell cycle regulated, but the vast majority are, as well as all the other core cell cycle genes. So that's a little bit about the biology of the system. And I was saying earlier, we can interrogate the system genetically. So we have mutants that affect size control. There's this mutant in the RB gene that makes really small daughters, and in the DP gene that makes daughters really large. These are size histograms. These are both encode conserved proteins that are part of chromatin binding protein and sometimes transcription factor, the RBE2F complex. It's the canonical idea in the literature is that this transcription factor turns on transcription of cell cycle genes. And I just showed you that there is cell cycle controlled transcription in Chlamydomonas. To our surprise and somewhat consternation, we see no evidence that this complex in Chlamydomonas has anything to do with cell cycle transcription, though it clearly controls the extent of cell cycle progression and that sizer mechanism. So one of the ideas we're entertaining and trying to test now is whether there's some direct impact on chromatin, not necessarily through transcription, that alters the cell cycle. And the idea is that maybe this complex, which we know is bound to chromatin, is affecting histone and chromatin conformation. And so the kind of questions we want to be able to ask are, first, how do Chlamydomonas histone PTMs change during the cell cycle? This is a kind of a summary of what's known in Arabidopsis about histone and chromatin changes over the cell cycle, but we really don't have this kind of information for Chlamydomonas. And then the second kind of question we're trying to answer is, does this retinoblastoma complex in Chlamydomonas have any impact on histone post-translational modifications? And so we really want to be able to answer both these kind of questions. So just a reminder that histones form an octamer, and octamers are made of dimers of 4 subunits, H2A, B, H2,3, and H4. These are really, really highly conserved proteins in their core region that form this octamer, and also in their tail regions, the N and C termini that stick out. And it's these N and C terminal regions that are often decorated with multiple kinds of post-translational modifications. And the idea that's been around for quite a few years now is that there's a kind of code to these modifications, that there's specific combinations of different histone PTMs. And if you want to interrogate what they are at a specific time and place in the cell or a specific cell type, or under a specific condition, if you did it by bottom-up proteomics, you would miss all the connectivity, the relationships between individual PTMs. And so doing traditional mass spectrometry doesn't work well to answer the kind of questions that we want to answer. And so working with EMSL and our other collaborators, we have spent the past few years and are continuing to work on this, developing new and improved methods to do top-down mass spectrometry on histones, where we look at the whole protein and analyze its PTMs. Our first manuscript on this was published earlier this year, and there's more work on the way. I don't want to go into any detail other than that. Working with EMSL, we were able to massively expand the number of known proteoforms or kinds of histone PTM combinations in the 4 core histones and linker histone in Chlamydomonas. So already this gave us a lot more information than we had before. The details in here are not very important. I'll give you a little bit of a more detailed example here of what data looks like. So the histones are purified and then put over a chromatography column and eluted into the mass spec. And the chromatography, first of all, does something very nice. It allows us to separate the oxidized versus unoxidized histone forms. This oxidation is an almost unavoidable byproduct of the purification process and changes the mass. We can completely separate the two. The y-axis here are the different masses, and you can see abundances and this laddering of masses. The higher up it is here, the larger the mass. And this is histone H3, and it's been bracketed out and separated for 2 specific post-translational modifications: a single methyl group on this lysine 4 and trimethylation on this lysine 4. And this is of interest to us. It's not only telling us what PTMs are there, but also we can do quantitation on them. About 85% of the total H3 is monomethylated and has low mass. It's got some additional acetylations in other parts of the tail, but they definitely have this known methylation, the single methylation. And about 8% of the histones have trimethylation, and again, a larger constellation of additional PTMs elsewhere on the tail, and we're working on deciphering what these are. And this is biologically interesting and relevant. I'm showing you, whoops, I just showed you, and I have to go back because I skipped forward too quickly. This is a ChIP-seq experiment for trimethyl K4, and it's in a very discrete set of peaks around the promoters of genes. And essentially what we're finding in this data, which is a collaboration with the Sabia Merchant's lab and JGI, is that this methylation mark, for example, is on a very discrete set of histones, probably a single histone at the promoter of each gene. And that about corresponds to the 5 to 15% of trimethylated histone H4 that we see in bulk, and essentially the rest of the histone H3 in Chlamydomonas is monomethylated, and that's found everywhere else. So this is just our kind of first toe dipping into the water for histones in Chlamydomonas, and what we're doing now is analyzing data from synchronized samples where we collected and purified histones from wild type at different stages, rather than just bulk histone analysis on asynchronous cultures that we did before, and also histones from mutants in this RB complex. And so our ongoing work is cataloging what histones change during cell cycle transitions and how they're affected in this mutant, the original questions we wanted to ask. I think the next slide that shows up just shows again that some of even these histones we saw asynchronously do change somewhat dynamically during the cell cycle, and having the quantitative information and the ability to quantitatively analyze this gives us additional information that we don't get from simply the bulk asynchronous data. So this is our in-progress work. And just as a quick summary of the top-down mass spectrometry of Chlamydomonas histones, the top-down mass spectrometry is really well suited to histones because they're very small proteins and because the post-translational modifications on them are both complex and combinatorial. The Chlamydomonas and algal histone PTMs are really relatively unexplored, and our collaborator, Jim Pesavento, has already helped discover a new PTM that's not found in plants or animals or fungi, but is found in algae, this histone H4K79. We're very interested in what the biological function and significance of that PTM is. And Chlamydomonas cell cycle controlled histones is a relatively unexplored area. And then finally, the retinoblastoma complex may control chromatin and even cell cycle progression through these modifications. And so the biology that we're discovering through mass spectrometry, we think will be very relevant to how cell cycle control works in this organism and maybe other organisms. So I think it's now going to switch over to Mowei.
Something. Can you hear me?
Yes.
Yeah. Somehow I cannot show my camera. The control is gone. Oh, okay. Oh, I see. Okay. Sorry. So I'll just be briefly talking about a little bit of technical details on the native mass spectrometry side. I'll be highlighting 3 recent examples working with EMSL users. So I've heard from Lily that the native mass spectrometry is focusing on looking at the high-order structures of non-covalent protein complexes. So the major difference from the technical side is that normally, historically, people have been mostly doing the protein work with denaturing conditions, meaning that although we start with the electrospray ionization, we use denaturing solvents, including organic solvents like acetonitrile and some acid in the solvent. And in this case, we see the proteins are usually denatured, highly charged, and this makes it easier to detect with the mass spectrometry because the mass charge is at a relatively low values, and it's easier to manipulate those ions. But with the development of the technology and instrumentation, we can actually reach to much higher mass charge with the modern mass spectrometers. And if you use non-denaturing solvents such as ammonium acetate at pH 7, when you do the same experiment, the same sample, but with a different solvent, you'll see a very different spectrum in this case for the same C-reactive protein, CRP protein. And this basically shows the mass correspond to the pentameric species of this same protein. And this is indicating that we are preserving the non-covalent interactions and the protein complexes in the gas phase. And we can do a lot of interesting mass spec experiments on them, and including getting the stoichiometry and also trying to understand the ligand binding properties. One sec. Okay. Okay. Yeah. So because we can measure the mass very accurately, we can easily look at the ligand binding to proteins, and we can resolve like 1 ligand binding or 2 ligand binding events based on the mass spec measurement. We can also quantify the relative abundance of those different species, the apo versus different bound species, and to read out the affinity. And those experiments have been very well established and in the literature, and have been widely used to study binding of ligand to or metal ions to proteins. So in addition, just like what people have been doing mass spec for peptides, metabolites, or lipids, the protein complexes can also be activated and fragmented for getting more structure information from the same experiment. There are a lot of different techniques, which I won't go into much detail, but some of them are more focused on collision with either gas molecules or surface target, and also there are electron-based activation and a photon-based activation that can break the proteins in different ways. So some of them are more focused on breaking the non-covalent interactions And in this case, we'll be able to get some compositional information, stoichiometry, and the connectivity of subunits. And this is more in the realm of quaternary structure protein complexes. Some other methods are better at breaking the protein backbone, and this will allow us to understand the sequence to identify the proteins, and also start to probe the secondary and tertiary structures, because the efficiency of fragmentation tend to be related to the structure, the higher order structure of the proteins. So just a quick overview. For native mass spectrometry, it's a very fast experiment and takes like minutes or like less than an hour to do a lot of the experiments. And it's very versatile because we can actually change the experimental conditions slightly to be able to analyze both soluble proteins, membrane proteins, and also intrinsically disordered proteins. And it's because the mass spectrometry can resolve very different species, it's compatible with heterogeneous samples that may not be easily resolved by conventional methods. And it's highly complementary to other structural biology tools such as cryo-EM, crystallography, and NMR, which I'll show a few examples in the next few slides. There are some limitations still because it's an emerging technology and it's still being developed. So right now, it's not the best to deal with very, very complex samples at the system level. We usually need some purification, or it can work with recombinant proteins. And it does require some specialized instrumentation for the best information. So yeah, so for the 3 examples I'll be showing you are all like work with EMSL users, and they're published in recently, and they're all very big collaboration efforts, including Professor Thibault's group and Professor Hellman's group, and also Professor Lewis's group, and some other folks at the PNNL and also other institutions. And the 2 studies shown here also in close collaboration with Professor Vicky Wasaki's group at Ohio State University, which is an NIH-funded national resource for native mass spec-guided structural biology, because we're really trying to apply the new technologies and instrumentation to be able to solve those challenging structural biology problems. So to start, I'll start the first example, which is the collaboration with Professor Thibault's group at OHSU. And this is about a study of a bacterial biomineralization enzyme, which we call it MINX. And so Professor Thibault's group discovered some marine bacteria in those environments that are responsible for biomineralization process. And this is a very important process involved in global biogeochemical cycles of elements. So they were able to identify this bacterium, and also they were able to identify the enzyme that was involved in this manganese oxidation process. And they were able to recombinantly express the protein and into active forms. And on the native gel, you can see when you add manganese 2 or manganese 3, you will be able to see the manganese oxide minerals forming, as shown by the brown color. What's interesting is that this protein is actually not a single protein, but it's actually a protein complex. So they have to co-express several genes to be able to get an active complex, active enzyme. So the protein complex contains a protein subunit MINX-G, which is a big protein about 138 kilodalton, and it has a lot of homology to other copper oxidases. But what's interesting is that they also have to co-express the 2 small proteins, MINX-C and MINX-F, and there was no homology to known structure for those 2 proteins. And Professor Thibault's group have tried a lot of times on trying to get crystallography data, but it's hard to crystallize. The protein complex is about 200 kilodalton, which is too big for NMR, and also it's kind of on the smaller side for cryo-EM. So we started this collaboration by using native mass spectrometry to help understand the structure. So we electrospray the protein complex recombinantly made, and then we activate the protein complex to break it into smaller pieces. And those are the raw data. You don't have to spend too much time in it. But the idea is that we can use very specific ways to break it, either collide with surface or collide with gas. We get different spectra. And it is very important to be able to break it in very specific ways. Like in this case, the surface collision actually allowed us to get a lot of different species. And then this fragmentation information helped us piece back the original structure of the protein complex. And from this data, we knew that the protein contained 6 subunits of the smaller ones, 3 of each. And they're proposed to arrange in this alternating fashion. And this hexamer of the smallest protein subunit will bind to the big subunit. At the same time, because we can resolve the masses of all those peaks, if we look at the masses of the two small protein subunits released from the complex, we detect different metal binding, the copper binding on those two subunits. And this gives us clues that the protein subunits probably have different functions in the process because they have different metal binding affinities. With this kind of information, we can start to build models without having high resolution structure. And we came up with a very plausible model that satisfied most of our experimental data. And this is work in progress with my colleagues, cryo-EM colleagues at the EMSL, and also with Professor Thibault's group, trying to really get the high resolution structure with cryo-EM. But it has been very difficult because there are some special issues of getting very good data from cryo-EM. But this example really shows us that with the native mass spectrometry experiment, this takes probably less than an hour to perform everything and get the data analyzed. And this gives us very quick measurement on the structure of the protein complexes without having to have very high resolution data. So the next example I'll be talking about is the plant pseudoenzyme enzyme complex in vitamin B6 biosynthesis. The pathway of the biosynthesis is here, which involves two families of PDX proteins, PDX1 and PDX2. So in this structure study, we're particularly interested in the interaction with PDX 1.2 and 1.3 in this family of proteins. So PDX 1.2 is actually an inactive pseudoenzyme that's very homologous to the active enzyme of PDX 1.3. So they have very high sequence identity and also homology. PDX 1.2, although it doesn't have its own activity, is known to positively regulate PDX 1.3. So the mechanism of this interaction is unknown. And PDX 1.2 has been very difficult to crystallize, but my cryo-EM colleagues was able to actually solve the structure by cryo-EM. And they both have the similar structure of dodecamers of two stacked hexamer rings. But still, we're still trying to understand this hetero interaction between the two proteins. There was some studies proposing that maybe those two proteins will interact in this kind of hetero complex, one hexamer from each, but it still needs to be experimentally tested. So to do this work, if you were at the previous EMSL seminar in October, my cryo-EM colleagues, Irina and James, they showed the cell-free expression system that's available in-house at EMSL. And we use this system to be able to express the different proteins, including the HOMO 1.2, 1.3 complexes. And those are the gels, the denaturing gel and native gel, showing we are able to express those HOMO proteins, which we only see one band, which is expected. And we can also use the cell-free expression and mix the DNA at different ratios to make different combinations of the hetero complexes. And at the same time, we can easily do some activity tests to make sure that those proteins behave as what they're supposed to be. And you can see that the ratio of the two proteins in the co-expression can be easily tuned. So with native mass spectrometry, what we're doing is that we can easily measure the stoichiometry of all those proteins we expressed. So the HOMO complex, you can see the data here. This is the raw data. You don't have to spend too much time on it, but this is the deconvoluted mass of the same data, which has an easier representation of the stoichiometry of the species in the samples. And you can see that we get mostly homogeneous species, but with the hetero complex, we see a lot of different species corresponding to different combinations of stoichiometry. This tells us that the protein complex of the two proteins does not have a very well-defined stoichiometry. Instead, they are mixing up with very different ratios depending on the availability of each of the protein subunits. So after we knew the stoichiometry, we still want to get a high resolution structure, but it's very, very difficult because, as you know, we have very different mixtures of stoichiometry of the hetero complex. And again, at the same time, the PDX 1.2 and 1.3 are so structurally similar, as shown by the monomer structures overlaid here. They're very difficult to differentiate in the crystal structure or the X-ray, crystal structure or the cryo-EM data. So, but if we use native mass spec, again, because each of the hetero complexes have different stoichiometry and they can be mass isolated in the mass spectrometer. And then if we break those hetero complexes apart, we release the hexamers in the lower mass region, and we can actually also measure the stoichiometry of the hexamers. And this is very useful because it tells us something about the composition and arrangement of the subunits in the heterocomplex. So to summarize the data at a higher level, if we do this experiment or for all the different stoichiometries, we see that the hexamers released from the complex is highly selective. That's almost about 1/2 of the stoichiometry of the dodecamer. This means that there's a higher level of symmetry when the proteins are assembled. So what does this actually mean biologically? So we also look at the surface charge in the homo complex versus the hetero complex. And this shows the homo complex of 1.3, the active enzyme, and the substrate is the binding site is here, shown by the yellow molecule. The hetero complex shown on the right. If you introduce a PDX 1.2 monomer in the hetero complex, it significantly changes the surface charge to negative values near the active site. And we propose that a mechanism of supercharging because of the 1.2 that will actually promote the release of the product by charge repulsion. And this explains the positive regulatory effect of PDX 1.2. And with all the experimental data, and including some of the data I didn't have time to explain, but the current model we propose is that there is some higher level symmetry when the proteins interact, and it will form this hetero complexes that maximizes the hetero interaction. And this will induce the maximum effect of the positive regulatory effect. And that explains our experimental observations. And this example shows how versatile the native mass spectrometry can be to really look at the heterogeneity of protein complexes that cannot be easily resolved otherwise. The last example I want to quickly mention is this cell wall proteins, digital protein discovered almost 20 years ago by Professor Roman Luis's group. And it has been indicated to be involved in lignin formation. And as you know, that the lignin can have a lot of different properties depending on the chemical and physical properties of the lignin polymers. And what's interesting is that the digital protein discovered, it can regulate the stereochemistry of coupling of phenolic compounds. And this digital protein discovered was able to control the stoichiometry of the product of coniferol alcohol when it dimerizes after treatment with oxidase. And if you don't have this enzyme present, you are getting a racemic mixture of the products. But if you have the enzyme present, you have very selective stereochemistry of the product. So they discovered that protein 20 years ago, but because this plant didn't have a sequenced genome, there wasn't too much study to study what was co-purified in this fraction and what other proteins may be important or maybe binding to the diligent protein. So we revisited this sample recently, and we purified the proteins from stem of the Forsythia plant. And we fractionated the proteins and used the mass spectrometry to study what we can purify in the active fraction. We also used we also used bottom-up and also top-down proteomics to be able to sequence the proteins de novo because we don't have the genome. With the help with some homolog the data, the transcriptomics and genomic data from close homologs, we're able to identify new proteins in the fraction. And we discovered new divergent protein homologs that's different from the original discovery. And those two homologs actually also form heterocomplex as reviewed by our native mass spectrometry data. And this example I want to highlight is because this really is where we want to go to be able to not only study the recombinant proteins, but also to study protein complexes that are extracted natively from the plants or other organisms. And this will preserve the native post-translational modifications and also potential ligands and other cofactors that are easily captured with native samples, and that may be difficult to study with recombinant systems. And with that, I just want to summarize the native mass spectrometry part for structural biology studies. So native mass spectrometry is a very fast experimental method for measuring the stoichiometry of non-covalent protein complex, especially shown by the example of the Minx protein, which we can get the data very fast and get some information, while it has been very difficult to get high resolution structure from the conventional methods. And the second example, the PDX example, showed that we can use those different activation methods to dissect the protein complexes to get more higher order structure information to help us understand the molecular mechanisms. And I would say that the native mass spectrometry really complements other structural biology tools for studying the heterogeneity of protein complexes. And that's all I have to say. And I think we're ready for Q&A.
Yes.
Wonderful. All right. Well, we haven't had any questions put into the chat window quite yet, but I do have some questions for you initially. So could you tell me what types of samples can we analyze with these methods?
Mowei
okay,
would be the best.
Yeah, I can take a quick on this. So as I mentioned briefly when I was talking about native mass spectrometry, so currently, like including what Jim was doing, like we're focusing on purified or partially purified proteins, and also we can easily do recombinant proteins. The complexity is still a relatively challenging problem to solve. But there are ongoing developments in separation techniques that will help us to directly look at the more complex samples. But right now, we usually have more like targeted analysis on a subset or a partially purified proteins. And I don't know if other people want to add something.
Yeah, I think that holds for both for top-down and native mass spectrometry, like in instance that Jim described, where we are looking at the histones. We do purify nuclei prior to enriching in histones. It's always useful if you can enrich subset of proteins or complexes that you are interested in.
Excellent. Well, thank you. What is the protein size limit of your technology?
Yeah, so I can also add on that. So for native, theoretically, there's not a real limit. The practical limit is more like if the protein gets to very, very big size, there are other techniques that may be an easier solution to start. Because once it's too big, it might be easier to do a cryo-EM. But the sweet spot, I would say, it's not like we cannot do smaller or bigger proteins, but the sweet spot is usually a few hundred kilodalton range for protein complexes for native mass spectrometry, where it is too big for NMR and it's kind of a smaller size for the easiest cryo-EM analysis. For top-down, I just like generally, we're looking at the proteins maybe a 30 kilodalton or a 50 kilodalton or less. That's usually the easiest to start with. And I know the histones are in the 10 to 20 kilodalton range, which are the easiest, the perfect range for doing top-down analysis. And I think other people may have things to add. I don't know, Lily, do you want to add something, or Jim?
Yeah, there are new technologies that are being developed to address this higher molecular mass proteins, including charge detection, single ion detection technology that will in not too far future allow us to look at the larger proteins and also more complex, more heterogeneous mixtures, such as what we encounter, for instance, in the example I tried to very quickly explain, where we are looking at the glycoproteins that are important for lignocellulose degradation, where we have extremely heterogeneous mixture, and the proteins are in the range, I would say, between 60 or 80 to 100 kilodalton or higher. So that's challenging, but there are technologies that will enable us to be more effective in that range soon enough.
Great. What is the throughput of the workflow? I mean, can we use it for large scale studies?
Yeah, so currently especially for native mass spec, it's mostly manual analysis, but we're in the process of trying to automate some of the steps, and that will help us to do like more higher throughput analysis, especially for smaller size proteins that are more within reach. For the top-down, the general workflow is similar to the omics pipeline at EMSL. Like the workflow is similar. the harder part is on the data analysis side, and I know some of my EMSL colleagues are also helping with that, trying to learn from what we have been manually doing, and then try to kind of automate that process to be able to do it in a much higher throughput. So right now, I would say the native mass spec is kind of in a similar manner as other structural biology work that's going on at EMSL, but it's actually a faster way of getting structural information. It's kind of, I would see it as more like a bridge between the omics data and the structural biology, and the throughput is kind of in between. The top down is generally in the standard throughput for the omics pipeline, but the data analysis is a little harder, but I'm pretty positive that will be improved very soon.
Great. What is the recommended process if I want to collaborate with you and use these methods?
Maybe Lily, do you want to take that one?
Yeah, sure. Yeah. So I think Linda mentioned that there are some active calls for proposal currently, but in any case, the best approach would be to maybe contact one of us or somebody else from EMSL that you might already have connection with, so that we can look at the problem that you have in hand and figure out what would be the best approach to that specific question that you have. And then we would work probably jointly on putting proposal of some type into the system that would then enable us, assuming the proposal is positively reviewed, would enable us to work together. We also can do other things. We are very open and actually very interested in collaborating with you as co-PIs on proposals to different agencies, to DOE, to BER, to NIH, if these technologies are of interest to you. So there are different ways in which we can collaborate.
Can I add something?
Yes, Jim, please do.
One thing I think that was really valuable for us is there are larger scale proposals that have to be fully formed and go over potentially several years, but we've really benefited from a smaller scale, more limited proposal that you can use to explore whether the method is going to be promising and give you the kind of data you need. And these could be just over a few months or a year. And I think it's a good way to, if you're trying to explore this, is to get your foot in the door. And it's of course easier to get resources for a smaller scale project like that than a full scale proposal.
Yeah, and the smaller proposal calls that they're talking about are the exploratory calls. Those you can find on the EMSL website. We don't have, it's not open at this moment. It will be open later on in this year, but that is an option for shorter, smaller projects. Okay.
There is, Linda, there is also, I don't know what's the name, but used to be called limited access proposals, so which are, I think, what Jim was talking about, which are, give you access to the facilities for maybe a month or two. So it's very defined scope where you can generate preliminary data for a larger proposal. And these are open. This can happen anytime through the year, but it's very much encouraged that you get in touch with someone in EMSL to lead you through that. Yeah.
Yes, you're right. The limited scope. In fact, for those who are interested in the different types of proposals that EMSL offers, here is a link in the chat to the webpage that has the entire listing of the different call for proposal opportunities. So to continue with the questions, how pure do the histones have to be for mass spec?
Jim,
okay, I guess I should be answering that. We initially tried to do some analysis with, I guess, partially purified histones and just using ion exchange chromatography, and it actually didn't work so well. So we needed one of the technical hurdles we had to overcome in this project was developing good histone extraction methods for Chlamydomonas strains that don't have a cell, that have a cell wall. The wall-less strains have been used for nuclei preps previously, but for various reasons, the science we needed to do had to be with a wild-type strain with a wall. And Dr. Pesavento, our collaborator, and his students really were instrumental in developing methods that could make histones where we could run them on just an idea of purity. I don't know the exact percent, but we can run them on page gels, and like at least half the protein there looks like it's in histone bands, and then there's a few other things. So for this application, we do need relatively good purity.
Great. And how do you purify histones?
Yeah, so it's a combination of a crude nuclear prep. We remove the wall, we beat them in liquid nitrogen, and then we do differential sedimentation to get crude nuclei, and then from there you can make use of the fact that histones are incredibly basic. So you do like a high acid extraction, and that leaves them in the soluble fraction, and that is more or less good enough. And then do a precipitation. That's more or less good enough to send them to Mowei to do the chromatography and mass spec.
Great. And it looks like we have time for one more question. And so that's how difficult is data analysis? Is it automated? I think, Mowei, you mentioned earlier that there is a certain amount of automation, but how difficult is it?
You want me to say something, or? I think Mowei has the most experience.
Yeah, so the identification of proteins is actually pretty easy. So the more difficult part is really to pinpoint the modification and know which site that is, and trying to accurately quantify the different combinations, especially for histones like the example Jim showed. Some of them, they are actually isomers that have the same mass, but the PDM can be at different locations. And that's the more challenging part. But like Jim Pesavento, like when we work with him, like he already has some papers published briefly with, we have some methods that can be used to help quantify the isomers based on the MS data. And then, but right now we have been starting this project with more like a manual analysis, partial automated with his script. I just want to make sure everything is working correctly. But I've been working closely with my EMSL colleagues and also Jim and Pesavento, also James Umen, and also Sarah. I've been working together trying to automate this with Yvette Belbo and also David Dagnan at EMSL, trying to put this into a script, and then we can then just load the data and just run it and then get a result, instead of people clicking through different software and copy pasting a lot of data. So
just as what you would do for the standard proteomics application, that is our goal.
Yeah.
Excellent. Well, our time is up. So thank you everyone for joining us today. We really appreciate your time. Don't forget to, you'll get a link to a post-webinar survey. Please fill that out for us. Your feedback is very valuable in making these EMSL Learn webinars better and better each time. So with that, have a great day. Thank you.